When a zebrafish senses the presence of food or a new sight or smell, its brain fires into action. The neurons in this fish have been engineered with a fluorescent gene that triggers small explosions of light whenever calcium ions produced by thoughts are activated inside the brain cell. And as the fish thinks, a state-of-the-art microscope developed by scientists at the Howard Hughes Medical Institute sends pulses of laser light through its brain. A camera records the neural activity from 4 different angles at 100 frames per second. The result is a 3D view of the fish brain at work.
We're interested in imaging basically the entire activity at the single cell level. So we can also then try to correlate information in different parts of the brain, see how different neurons might talk to each other.
Physicist and biologist Philip Keller developed the technology. Ultimately, he and his research partner, neuroscientist Misha Ahrens, would like to see how the human brain functions in fine detail, but the technology doesn't yet exist. Keller's microscope, however, is at the cutting edge.
The special feature, the special concept in this system is that we can image very quickly a complex, large sample, large 3-dimensional volume, and we can actually look at that volume from multiple different perspectives at the same time.
Those multiple perspectives, or sheets, are combined by a computer program to form a single moving 3-dimensional image.
Because everything but the sheet is essentially invisible, we can capture what happens in the sheet that's being excited very accurately. And through molecules that report neural activity, we can then essentially see what these neurons are doing at the time that we're filming them.
And the technology is not limited to recording the activity of zebrafish brains. Here, in a compilation of millions of images recorded over 20 hours, a cluster of cells evolves into the elongated body of a fruit fly embryo, with muscles forming moments before it becomes a fully formed larva.
One typical experiment we would do with this microscope, and that is not possible with a different imaging system as they existed up to this point, is to really follow basically all of the cells in a developing embryo. So we start as early as possible, so go ideally back to the 1-cell stage at the very beginning of the developmental process. And then we try to follow these cells through development, through the entire phase of embryogenesis, see where the cells end up, how they move, how they interact, how they divide, in order then to build this complex structure, the complex multicellular organism with many different types of tissues and organs that have quite impressive functionality. So you want to capture that entire process as it happens for the entire living system.
The imaging technology is improving. For brain mapping, transparent zebrafish are ideal models and provide crucial information, although Misha Ahrens says he's hoping for more.
We see a signature of neural activity, but we're not yet seeing the very fine structure of it. So in the future, we're hoping to speed up the system so that we can see individual spikes and we can really tell exactly when one neuron sends an impulse to another neuron.
The ultimate goal is to study neuron communication in human brains and better understand the nature of neural disorders like Alzheimer's and Parkinson's disease, so more effective treatments can be developed. It's a long way off, but through scientists like Philip Keller and Misha Ahrens, the groundwork is already being laid.